So welcome everyone again. My name is Allyson Berent. I am the Chief Science Officer for FAST, obviously the Foundation for Angelman Syndrome Therapeutics. I'm the co-director of the ABOM, the Angelman Syndrome Biomarker and Outcome Measure Consortium. I'm also the co-director of InSync, the International Angelman Syndrome Research Council, and I'll talk all about these things today. I'm a veterinarian, I'm a clinician and a clinical trialist in animals with naturally occurring diseases, and that's where my career certainly started. And the most important job that I have is I'm the mother to 3 beautiful little girls, and our middle daughter, Quincy, is 8, and she's there on the right of the screen, and she lives with Angelman syndrome. So I want to just direct you to this pamphlet that you have on the table. This is really meant to make this talk simplified, because I know that so many people give really incredible feedback about this talk and how you really love to understand and learn the science. And you look forward to this every year, which is the most humbling feedback that I get of how much you want to learn and know and understand. And the fear is that it makes sense for the first 5 minutes after I speak, but then you might forget what I said. And so I think, as I said earlier, knowledge is power, and you need to be able to walk around in your community and reiterate everything in here. You need to walk up to the school teachers of your child and explain to them what an ASO is. You need to explain to them what gene therapy is. You need to explain to them why a child might be walking around with little devices, little sensors on their ankles, because we're looking for outcome measures and biomarkers and what that means and why it's important. And if you can embrace it and understand it and own it, then you are going to be the best advocate for accelerating drug development for Angelman syndrome. And this is an army that we need to be able to do this around the world. This can't just be coming from the organization, but it has to be coming from you. And so I'm here to educate you to be an ambassador for your family, for your child, and for our community. So I hope that this is very helpful, and this will follow along in my talk as well. And so I'm going to reference some spots in here as I dive deeper into some of the scientific details for you to be able to kind of take it back to the basics when you walk out of here and forget everything that I just said. So for my disclosures, I am the co-founder and former chief operating officer of Genetics Biotherapeutics. That was recently acquired by Ultragenyx. I'm a consultant for Ultragenyx. I'm the chief development officer for Mosaicism Therapeutics, which is unrelated to Angelman syndrome. And I'm a consultant, and I advise many developmental initiatives for different neurodevelopmental disorders in many organizations around the world. I'm also a member of the N of 1 Collaborative for rare diseases. And so our roadmap. So Craig just showed a lot of our roadmap that really got grown and accelerated in 2016, thanks to an incredible gift from the Meunier de la Postal Foundation. So Julian is here in the room, and his family, who really absolutely, oh, I know you're embarrassed. You're in the dark. There's no light on you. You're okay. But they ignited drug development for Angelman syndrome, and I think none of us can deny that. And if it weren't for them, we would not be where we are today, and there's nothing around that. But what I can say is that we're now in a place, thanks to genetics and the Ultragenyx acquisition, that allows us to be able to go to Roadmap 2.0. And we're able to do that because this infusion of funds has allowed us to be able to grow, to advance, and to take the vision and the mission that we've always had and make it turbocharged, because that's what our kids need and that's what our kids deserve. And I hope I can share a little bit of how we can do that today, because I think it's incredibly important. So I'm going to go back to the basics, because everybody asked me, please go over the basic genetics of Angelman syndrome again. Don't lose that. We need to understand it. So here we go. And I'm going to do this relatively quickly, but I want to be sure that everyone understands, and again, ask questions. So if you all remember, almost every cell in the body has 26 pairs of chromosomes. Okay, so that's 46 chromosomes. We have one from mom and one from dad. And on those chromosomes, there are genes, and those genes are read and make proteins. And Angelman syndrome is caused by either missing or a non-functional copy of a single gene on chromosome 15. And unfortunately, as we all know, that single gene is incredibly important. It affects potentially 1 in 15,000 people around the world, which means that there should be 500,000 people identified globally living with Angelman syndrome. And we're going to talk a lot more about that this afternoon when we talk about the Search and Rescue Initiative. It's random, it's rarely inherited, but it can be. It's considered a de novo mutation in that usually parents are not carriers for this. Each chromosome, like I said, is made up of these various genes that code for various proteins, and those proteins allow our body to function the way it does. And so when we look at chromosome 15, I'm going to make this a schematic just to make this a little bit less complicated. And we look at the cells of the brain, the neurons in the brain. This is what it looks like. So we have a mother's copy called the maternal, and we have a father's copy called the paternal. And normally in all cells of our body, they're both red. And so we can read both copies. And if we only need one, it doesn't really matter because we have the other one, because they're almost identical in how they function. But in the brain, for some reason, which I'm not happy about, someone decided that it was a good idea to silence the father's copy of the gene. And that's called imprinting. And there's very few genes in the world that are imprinted. Scott Dindo's actually an expert on imprinting disorders. And so he could talk to you all about that. And he feels like there are very good reasons for that. I think there's a really bad reason for that. It's called Angelman syndrome. But ultimately, it is imprinted. And what that means is that that copy has this long strand of RNA sitting on top of it, which is genetic code. And it is stopping the translation or the reading of the UBE3A gene on the father's copy. And that's in all of us, in me, in you, and in our children. But why don't I have Angelman syndrome? Because my mother's copy is being read, and we have that UBE3A gene right here on the mother's copy. But Quincy doesn't. She's missing that whole segment and does not have a readable mother's copy. And therefore, she doesn't have any functional copy in her neurons. So she has Angelman syndrome. And that is really what you need to understand in order to grasp what we're going to talk about for the rest of the weekend. Because if you don't understand that, knowledge is not your power. Where are these drugs working and how are they working? And how am I making decisions for my child who may have a different genotype than your child? And we need to make those decisions for our own children. And so there are many genotypes of Angelman syndrome. Here's an example of deletion. So in a deletion, we have a piece that's missing. So it's shorter than everybody else. In a mutation, we have a letter that's been misread. A T is not a T, a G is not a G. It's not read properly. And therefore, the protein is not being read. And then there's UPD and ICD. And those behave relatively similarly in that there's two fathers' copies. They're both silent. They're both healthy. The gene's there. Can you believe it? It's two healthy genes there, not one healthy gene there. And they both happen to be silent. That's unfortunate. But that's important when we think about what are the options for therapies for all of our kids. And then an imprinting center defect is similar. And we'll talk a little bit about mosaicism, which is even more rare. So we have really common deletion. The mutations are a little less common. UPD and ICD are even less common. What does UBE3A do? Well, it is a protein that's very important in neurons. And what it does is it actually tags other proteins in the brain and in the neurons to function, to be degraded, or to be upregulated. So it's saying you have too much of this and not enough of that. And I need to knock you down and I need to raise you up. And we need to have this balance of all the proteins in the neurons so that the two neurons can talk to each other. And that talking happens at this area called the synapse, which is the junction between two neurons and how they communicate. And so this gene called ubiquitin ligase is an enzyme that allows different proteins in that area to either be broken down or not broken down. And therefore, if you don't have UBE3A, you have what you call a synaptopathy or a problem at the synapse. And what is that problem? That problem is that we don't have enough of the calming proteins called neurotransmitters, like GABA genes, GABA neurotransmitters, but we have a lot of the excitatory neurotransmitters. So that balance is really off. And what does that mean? That means that we don't have enough what we call tonic inhibition. This community loves the word tonic inhibition. Use it all day. Just go around and talk about tonic inhibition. You'll sound brilliant. But what that means is that we can't coordinate relaxing. So if I go to walk and I take a step, I activate one leg and I inhibit or I relax the other leg. And it's a coordination of excitation, relaxation, excitation, relaxation. If you only have excitation and you don't have relaxation, what happens? Well, unfortunately, Angelman syndrome happens. And so this is what we're left with. We're left with the symptoms of excitation, which is a lack of speech. Why? Because you need to relax and coordinate all the muscles of your tongue, your voice box, your face, your cheeks, your head in order to speak. And if you have dyspraxia and apraxia and incoordination of your muscles, the thought of how you're going to coordinate that is impossible. And so it doesn't mean our kids don't have the ability to speak. It might mean that they just can't because motorically they are challenged with overexcitation and lack of inhibition. It also might mean that the brain that's trying to get the communication, not only to functionally motorically speak, but to think about how to speak, is really, really noisy. So all this excitation, I always say, is like you're driving in a drizzle, and then it becomes a rainstorm, and then it becomes a thunderstorm, and then it's a downpour, and you have to pull off the road because you can't see, because the windshield wipers are going like this, but the rain is so hard and there's so much noise. So what does that mean? That means that it's really hard to function and see through the rain, because that's a lot of noise. And that's what our kids live with every single day. Life for them is like a really bad rainstorm, and they're trying to see through it. And frankly, they're functioning pretty darn well for how hard it probably is for them. And so basically they have a lack of speech, debilitating seizures. They have a hard time coordinating, and they could fall a lot, or maybe even lose the ability to walk, or never gain the ability to walk. And probably one of the most profound things is they can't sleep. If you can't relax, you can't sleep. If they can't sleep, you can't sleep. That's not great. And so all of these things are for this single gene. And I don't need to go through with you the symptoms because you live them. But what about the impact it has on families? What about the impact it has on you and the caregivers? Some people can't maintain employment, significant anxiety and depression, stress, social isolation, loss of sleep. Big problems that we live with because we are caring for these children that really have quite a few challenges. Doesn't mean they're not amazing, but they have a lot of challenges. And so the deletion is a little bit different in that the deletion kiddos, like I showed earlier, they have this segment that's missing. And when we break that out into a schematic, what you can see is they're missing genes outside of UBE3A, and they're missing these GABA genes. So they could be missing a whole segment, which could be up to 12 or 15 genes. The most important one is UBE3A, but that's why deletion phenotype, the behaviors of kids with deletions, often marginally more severe than those with mutation or UPD or ICD, because they're also missing these other genes that probably have a major impact on tonic inhibition. So that's a segue to remind you that tomorrow there's an excellent talk that's going to be going over a new drug, a new clinical trial that's going to be launched by Roche. And they're going to be talking about this tomorrow at 10:45 a.m. So make sure you're there. Make sure you circle your agenda for tomorrow, because they're going to talk about a new drug that's going to be targeting this area in order to think about how they can improve deletion patients outside of UBE3A. So when Quincy was diagnosed, this is one of the most important papers that I ever read. And it probably is what got me to this stage, because what I realized when I read this paper was that there is so much hope for our kids. So there's a very small subset of kids that have something called mosaicism, and mosaicism is incredibly rare. There are people in this audience whose kids have mosaicism. I know that. It's really rare, and it's so important. Why? Because what it told us, besides that your kids are awesome too, what it told us is that if there are kids that have only 1 to 5% of cells that are expressing UBE3A, so mosaicism means some cells are expressing it and some cells are not. They don't all have it missing. So there are certain numbers of cells that have it and don't. And so if 1 to 5% of cells are expressing UBE3A, then you have no to few seizures, you're fully ambulatory, maybe have some ataxia, and you have some speech. 1 to 5% expression is a different child than Quincy. Different. Imagine that. 20% fully ambulatory, speaking in sentences. Can you imagine if that was our life, the impact that would have on our kids? Kids that are diagnosed with mosaicism, over 40% are not really reported. Why? Because they're probably neurotypical. So you probably do not need more than 40% to be considered significantly delayed enough to get a genetic test. Can you imagine if my kid didn't really need a genetic test because it's not really clear if she has a genetic disorder? That would be profound. So ultimately, what we know is 1 to 5% is something that we would take, because that would be a different child than what most of us live with every day. And 20% would be absolutely glorious, because that, again, is a child that can live a more independent life than what they can today. So we're going to go through 4 pillars, which is the pillars are here in your handout. And the 4 pillars from a schematic perspective are pillar 1, fixing mom's gene, right? We can just replace mom's gene. We can fix it. We can put it back in or put the protein back in. Pillar number 2, we're going to turn on dad's copy of that gene. So we have this silencing. How do we unsilence? Pillar number 3, we're going to address the synapse. We're going to address the junction of the 2 neurons so that we can think about how downstream targets are impacted, the GABA receptors, how those receptors are really approached, and how we may be able to help with some of those symptoms. And then pillar number 4 is how do we accelerate for pillar number 1, 2, and 3? What do we need in order to not only get that to animals and developed, but actually to patients and to approvals? What is it going to take? And so this is very, very important. But I'm a scientist, so I'm going to talk about the pillars in reference to neurons and viruses, because that's the way that I understand drug development the best. So pillar number 1 is gene therapy, replacing the missing gene or protein. Pillar number 2 is unsilencing. And pillar number 3 is downstream targeting. And pillar number 4 is preparing. We need to prepare for clinical trials. We need to prepare for drug approval. And so when we think about these modification strategies, thanks, Craig, for stealing my thunder, what is important to think about is how are we going to get the gene in there? How are we going to get the protein in there? Okay, so gene replacement therapy or protein replacement therapy can be delivered through adeno-associated viruses or through lentiviruses using hematopoietic stem cells, or through enzyme replacement therapy. And these are all the people and academic collaborators working in that space. This is a microglial cell releasing UBE3A by hematopoietic stem cell delivery. And this is an adeno-associated virus in the brain delivering UBE3A by secreting it. We can stop the stop. We can activate the paternal copy of that gene. And we could do this through various different mechanisms which have the same concept, but just a little bit of a different technology, a different platform. Or we can do downstream targeting, and we can really use drugs that are really going to address that synapse. But I'm not going to talk about that today. Dr. Cheryl Fife-Marchich is going to talk about that right after me, and she's going to talk about the basics of gene therapy and ASO therapy and lentiviruses and adeno-associated viruses, so that you can really understand 8th grade science class is what does this mean and how does this work? And I think it's really important for us to understand that. So let's just focus a little bit on pillar number 1. Again, I'm not going to talk about the details of the science of gene therapy. I'm going to talk about the details of what's happening in Angelman syndrome. Pillar number 1, this is really what you need to know. So after gene replacement therapy, and this is with a hematopoietic stem cell gene therapy out of UC Davis, what you can see with this mouse on the left is this is an Angelman mouse who is having a real hard time keeping up, having a real hard time coordinating his front feet and his back feet. I'm sorry, but it does look a little bit like Quincy. It's hard for her. It's hard for her to keep up on a treadmill. It's hard for her to run. It's hard for her to not be hit by the backstop, right? But this is that same mouse after gene therapy. Suddenly, this mouse is coordinated, can run, can walk, can keep up, is not hitting the backstop, and is able to keep the speed of a wild type normal mouse. And so that's where you start in drug development. You need a proof of concept, and you can't see a more clear proof of concept than something like that. So these gene therapy programs, I'm not going to go through the details, but I just want you to see how much work in science has been done over the years. University of South Florida in 2011 was the first to ever publish on gene therapy in Angelman. Thank you to Dr. Weber, who really took the lead and said, I think we could do this for Angelman syndrome. And since that time, many other programs like UPenn and University of North Carolina, Dr. Philpott and Dr. Jim Wilson are doing similar but different strategies. Hematopoietic stem cell gene therapy, Joe Anderson at UC Davis took this approach and said, can we deliver this through a different type of vector, a different type of delivery to get to more of the brain? What impact can we have there? And now there are 4 additional programs being actively pursued by pharmaceutical companies or academic institutions that are either undisclosed or not yet ready to publicize what they're doing. But there is a lot of work in this field right now. And today at 3:20, Dr. Wilson's going to talk about his gene therapy program. And on Saturday, Jennifer Panagoulias is going to talk about transformatics and hematopoietic stem cell gene therapy. And Dr. Cierlone Stephanie is going to talk about PTC's gene therapy program that was started out of Ed Weber's lab. So there's a lot that we're going to talk about that I just want to guide you through all of this today so you know what to circle on your agenda of what you want to go to. Enzyme replacement therapy is where you can actually just take the UB3 protein itself and deliver it directly into the brain through a reservoir that looks like this. And ultimately, there can be a reservoir that you have a depot of enzyme, you put it in the brain fluid, and then it actually can secrete into the brain. So we're working on that. And we're working to see, is that a possibility for Angelman syndrome? Is that something that can be done? It's been done for Batten disease. It's being done for other types of neurologic disorders. Is there a place for that for Angelman syndrome? And let's look at pillar number 2. What's going on in Angelman syndrome there? Well, we have a lot of different modalities looking at stopping the stop and turning on the father's copy. And that all started out of Ben Philpott's lab in 2011 when he took a chemotherapy called a topoisomerase inhibitor and found that drug activated the father's copy and turned it on. And that was where the whole field exploded because it realized that we could do this with things that maybe are a little safer than chemotherapies, but we could do ASOs, artificial transcription factors, and different types of RNAs that might be able to knock down that antisense transcript, as well as different CRISPR programs. And these are all the programs at work. And you're going to see this over and over again. This is for your notes, but I just want you to have it on a slide so you know all the things that are happening and all the companies that are working on it. So let's just dive quickly into antisense oligonucleotides. So this is the first to publish. This was Dr. Art Baudet at a Baylor College of Medicine. And what he showed was that in adult mice with Angelman syndrome, when he delivered an ASO, they had absolutely no UB3 being expressed. After delivery of an ASO, the brain was lighting up. It was on fire. Red is good. Red means UB3 is being expressed. So with an ASO, we have a lot of red. And not only do we have a lot of red, but we have behavior improvement in adult mice with Angelman syndrome. So this was proof of concept that there was a path forward for this for potential translation to humans. And now, after all that work, we have 3 clinical trials ongoing that started in 2020 between Ionis and Biogen, Ultragenyx, Roche and Genentech, and then C.U.R.E is working on something called an SNA, which is a little different, but the same concept. Dr. Kakis is going to be talking tomorrow at 9:45 on his program, Dr. Crean at 10:15 on her program, and Dr. Vicenzi at 10:45 on her program. So we're going to hear all about this in humans tomorrow. And this is all work that started in mice. And then what about artificial transcription factors? Well, this is something that Dr. Dave Siegel, who's sitting right here, presented at least to me for the first time in 2016, right after Quincy was diagnosed. And I was blown away. Why? Because there's a lot of red. I like red. And I saw all this red and said, that's good. That's UBE3A. We need to get that into these kids. And so he's done a tremendous amount of work with his team to try to figure out if there's a path forward for this for Angelman syndrome. And he talked a lot about this yesterday in the closed session with some unpublished data to show that there may be really still a path forward for this. And we need to do a lot more work to investigate that. But ultimately, this is another great approach that we need to keep our eye on and we need to support. And then finally, CRISPR. So CRISPR is gene editing. And gene editing is where the antisense transcript, just like an ASO, is going to be knocked down, but it's going to be knocked down permanently. So instead of needing a spinal infusion every couple of months, this is an opportunity where we may be able to do a one and done and deliver this so that we can knock down that antisense. So this is the antisense where it's very high, and this is how we can knock it down and make it lower. So ultimately, what you can see is that you don't need to go through this, but that these are mice that had phenotypic rescue. And Dr. Zilka published this in 2020 out of the University of North Carolina. And now there are many programs in CRISPR using Cas9, Cas13, a new Cas that was disclosed yesterday, and many new approaches that are being investigated out of UPenn, UC Davis, different pharmaceutical companies, and Yale, who's going to talk about their program later this afternoon at 3:55. And I just want you to look at this. This is the next most profound video I have ever seen. This is a normal wild-type mouse. This is an untreated Angelman mouse, and that's an Angelman mouse after CRISPR gene therapy. Look who didn't fall, the Angelman mouse after CRISPR gene therapy. Look how he's doing better than the wild-type mouse. He's a super mouse. And that little fella, I'm sorry, little guy, but you need a little gene editing so you can stay up on that rod too. And so this is proof of concept. This is what we fund. This is what we need to get to patients. And so finally, downstream targeting 3. There was a trial by Disruptive Nutrition on ketone esters. Neuron is going to be talking about IGF-1 agonist ligands. And then we just funded a new program out of Brown on a BDNF analog, Dr. John Marshall, who talked yesterday. And then Roche is going to be talking tomorrow about the GABA agonist. So these are all the downstream targets, and I'm not going to steal Neuron's thunder, but they're going to be telling you all about this tomorrow at 11:45. So tune in for all of these really exciting updates. And this is just a heat map. And all you need to know is I love heat maps. But green is good and red is bad on this map. Red is good if it's UBE3A, but red is bad on this map. So what this tells us is there's a lot of green and little red in both at birth or adult treatment. And what this gives me optimism for is that postnatal treatment for Angelman syndrome could have a profound impact because we could get rescue of a lot of the symptoms of Angelman syndrome. And I think that is incredibly important. So now how do we accelerate? How do we prepare for clinical trials for humans with Angelman syndrome? That's a lot of stuff and a lot of science in the background. But what about our kids? That's what really matters. And so there are a lot of efforts to support clinical trial readiness and innovative therapies. And I'm going to go through these throughout the course of the day. There's going to be talks on numerous things here. One talk today at 11:50 is going to be by Dr. Baker and Dr. Casera talking about newborn screening and how we can diagnose super early so we can potentially deliver to patients the day that they're born or within weeks of being born. That would be a dream. And we're going to get there. We're going to get there. We fund tools. As you know, we fund a rat model and a pig model that Dr. Dindo is going to talk about this afternoon, and a large deletion 5 megabase mouse model that we have the human counterpart in the mouse of the large deletion and all the GABA genes. And Dr. Yonghui Zhang, we funded him to make this model, and that model is now viable, and they're working to characterize that. iPSC cell lines for all of our kids. Every genotype has a cell line, and all of these genotypes are now being developed into these landing pads for therapeutic testing to see what drugs work best in what model. And so that's out of North Carolina State and Yale. And these talks today, Dr. Kyung and Dr. Bandekar are going to be talking at 11:10, and Dr. Dindo is going to be talking about the pig at 11:35. InSync, the International Angelman Syndrome Research Council, that's a group that we bring together from around the world in order to really advise fast and Angelman syndrome research. What are we missing from proof of concept to drug approvals? And we bring people outside of the Angelman space together to look at us and say, this is what you're not thinking about. This is an idea for Angelman. What can you do that you're not doing now? And we bring these people together once a year to really help us understand what initiatives we're not thinking about that are available. And so it's been an incredible, incredible ride to work with these great people. The natural history study for Angelman syndrome, we're on our 3rd version of the natural history study. It's been incredible. The first one was funded by the NIH, the next one by the FDA, and now this one is being funded by the ABOM in collaboration with the Angelman Syndrome Foundation, FAST, and multiple steering committee collaborators from the pharmaceutical field are all putting money in together to work together to support the Boston Children's Hospital natural history study, which is very exciting. The global registry will be further discussed today at 2:15. So I hope that you're there to hear about all of the patients that around the world that are registered in this registry. The ABOM, I'm going to be talking about the Biomarker and Outcome Measure Consortium at 1:15. So if you want to hear about endpoints and biomarkers for our kids and why they matter and how they matter, we're going to talk about it then. And ultimately, the primary goal is that each platform can benefit different targets, different individuals, and we need to de-risk each option for the potential for success. As Craig said, not everything is going to be successful. And we have to think about how we're going to test everything to have as many options as possible. Because what happens in drug development is for every 15,000 drugs tested in a lab, 5,000 might make it to the FDA type of testing that needs to be done before it can go into humans. Of those, 10 might make it into a clinical trial for one that might get approved. Are we willing to stop at 1 or 2 in the early phases if only 1 out of 15,000 might be approved? Absolutely not. We need to have all shots on goal to make sure that we can advance all of them. And 7 years ago, these were our shots on goal. 4 years ago, these were our shots on goal. 3 years ago, these were our shots on goal. 2 years ago, last year, we had 26 shots on goal. In March, we had 34 shots on goal. 3 months from this summit, we went up to 34 programs because people were in the room and like, I want to be part of Angelman syndrome. So it went haywire. And of those programs, these are the FAST-funded programs. And we robustly funded a huge amount of money into these programs to advance them. But unfortunately, after March, this happened, as Craig just discussed. The stock market went down for biotech. And what was the result of that? Those 34 programs had 9 programs pull out. And that went down to 25 programs over 2 months' time. But the FAST-funded programs not only did not get canceled, but 1 more got added. And so we went from 12 to 13 that FAST robustly funded, but we went from 36 to 25 that were in the space. And so what's super important here is that drug development is chutes and ladders. Okay, there's financial times that are bad. Science can be not what we expect. We need to pivot. We need to turn. We need to move a jet ski and not a barge. We need to be able to turn when we need to and advance when we need to and pull back when we need to. And sometimes it's not what we want. But if we're driving those decisions, at least we know the decision is never being made for a lack of priority, but is being made because the science wasn't sound enough to have a go decision. And so it's not only that drug development pipeline, but all the things below it that we need to get there. And so in 2015, we were working really hard in the discovery phase, improving diagnostic efforts, improving animal models. But where we are today is we are moving our road to the right. We are advancing all of the things we need to support not only getting to clinical trials, but we're in a position we need to support getting through clinical trials. And 3 years ago, I was not able to say that. We robustly advance and accelerate in drug discovery. And we are robustly and advancing drug discovery in humans and out of mice. And I think that is incredibly important for us to realize how far we have come as a community. And one thing that we realize is that we've funded over $30 million of research in over 20 academic institutions. And these pillars have gotten us across the line in order to support endpoints and community discovery and preclinical. But the thing that is so missing right now is not just preclinical and not just acceleration to humans, but now we have drugs going into humans. And the problem is that getting these clinical trials enrolled and activated is really challenging. Why is it so challenging? Because there's uneven efficiencies and execution of clinical trial enrollment. There are 4 active clinical trials right now for Angelman syndrome, 3 of which are disease modifying and 1 is a downstream target. Did you know that over 75% of the patients in the United States are enrolled at 1 institution? That is for 4 trials. What happens in the next 18 to 36 months when there are 8 to 15 trials opening, some of which are going to be phase 3 with 100 to 200 patients per trial? And we have some institutions that can enroll 1 to 2 patients a week. And we have some institutions that are only capable of enrolling 1 to 2 patients a year. How do we get hundreds of patients access to be enrolling in these trials and eventually drug approvals, where it's going to be 500,000 patients that need to be treated, if the inefficiencies of these clinical trial centers are not there? What can we do? And that's the gap that needs to be filled. And so how do we bring therapeutics to patients? We need to train clinical trialists. We need to teach clinicians how to effectively, efficiently, and expeditiously execute. We need to train people on understanding good good clinical practice. How do they dispense novel therapeutics that are complicated at a pharmacy? They're biologics. They're very complicated. How do we ensure that these trial activities are coordinated in a way that multiple patients can be seen a day and not a month or a year? How do we designate time for our best doctors out there that are working so hard and want to do this for Angelman so badly? What can we do to get them to open their slots so that they can see more patients and deliver these novel therapeutics so they have the time to be able to do that? How do we adjust their obligations that this becomes their obligation? Well, they need improved infrastructure. They need facilities and teams to be able to help them do this like a machine. They need space designated to run the trials and deliver the therapies. And that's different than general specialized care. This is clinical trials, which is incredibly different. And most people in their residency are not trained on how to run clinical trials. They need to focus on the needs of the patients during these trials. What are the needs of these kids? If you've ever gone to some of these centers, what you'll see is that you might be there for 8 hours. What is your kid doing for 8 hours? They're sitting in a hospital room or an exam room, and they're pacing the hallways because our kids don't sit in any room. So what can we do to make the experience better? They need sensory gyms. They need visual and textual friendly environments in order to be entertained. They need to be exciting. They need to be fun. They need to be looking forward to going to these places. They need a team to help execute contracts and ethics and service coordination. They need space so that all of the anesthesia teams, neurointerventionalists, neurosurgeons, neurologists, geneticists can all work together, neuropsychologists in the same team, that it doesn't have to be so hard to coordinate. And that's part of the problem with the delays that these sites are seeing. And we've talked to most of them. So we need centers around the world that can focus on novel, innovative interventional therapies for Angelman syndrome. We need to just really design a fellowship and an externship program that we can train doctors that have existing clinics that want to improve, or that are finishing a residency and want to become a clinical trialist. And we want to train them that their job would be running clinical trials for neurodevelopmental disorders like Angelman syndrome. We need world leaders to start these flagship centers in order to arborize and train the rest of the world on how to do this. And so when we think about things and gaps that we have, clinical trial centers for interventional and innovative therapies is something that's really missing. And so we've done it for mice, and we've done it for accelerating drug development through the pipeline of discovery. But now we need to accelerate drug delivery for people. And that is an amazing place to be. But fortunately or unfortunately, it also has an unbelievably incredible cost. So we figured out, how can we do this? How can we best utilize all of the funds that we're working so hard for to help our kids and to give better access? And after a lot of diligence and a lot of hard work, it's been a long time coming, but we're super excited to announce that we are finally able to launch our first FAST Center for Translational Research at Rush University in the pediatric neuroscience area. So Dr. Elizabeth Berry-Kravis is going to be the director of this center. The entire team is back there, this entire team is their entire team, they're all here. They're so committed to train everyone around the world to be able to function like a machine that Liz is. Liz is a machine that will see and enroll 2 or 3 patients a day if she could. The inefficiencies may not allow people to do that, but we're going to create a playbook so that we can know, what does the facility need to have? How does it need to be built out? Who needs to be hired? We need an anesthesiologist. We need an anesthesia room. We need a neuropsychologist. We need a neurobehaviorist. We need a neurointerventionalist. All of the pieces that need to come together to deliver these novel, interventional, complicated therapies. Gene therapy is complicated, but if you can treat 500,000 kids, you need to train people to do it. And it costs a tremendous amount of money to create this infrastructure. So we are committed to do this across the world. Over the next 5 years, we would like to open numerous sites around the world, but Liz is going to help us create this playbook. And this playbook is going to allow us to also train 2 fellows a year. We're going to train 1 nationally and 1 internationally, because we need these people to become clinical trialists in neurodevelopmental disorders and go out there and go to these institutions that we can bring infrastructure to and we can grow. And she will help guide them on how to become another machine. And if they can function at a level 10% of what she does, our kids will all get a treatment, because nobody will function like her. And I pray she lives forever, but she may not. And therefore, we need to replicate Liz Berry-Kravis. And so we are very, very excited to be able to build this team around her. And we hope that this is the map that we will show you one day that is arborized by starting centers like this, focusing on clinical trials and novel interventional drug delivery. This is not for holistic care, and this is not just for treating patients with Angelman on the day-to-day. This is meant to be within those types of centers for trials and for novel delivery of therapies, and really get these treatments to kids, which is absolutely missing. And this is something we are so very passionate about as part of our Roadmap 2.0. And so really, if you look at all of the things it takes to get drugs approved, it's expensive. IND enabling studies, clinical trials, getting through a phase 3, getting an approval, getting through the FDA review. All of that is where everything got pulled back because there was no money there. And this is where Alana and Ryan are going to talk to you at 4:20 about what does this look like from a business perspective on how we make sure 9 programs will never get canceled again if they have good science. We cannot let that happen. So our Roadmap 2.0, I expect to look like this in the very near term, where we have not only a couple of phase 1/2s, but we have drug approvals. We are in review. We are in phase 3. We have multiple clinical trial centers that are now becoming interventional innovative centers that are delivering these approved drugs at rapid speed so that every patient around the world, not just in the United States, but in all parts of the world, can receive the best care that our kids can get as well. And so this is so incredibly important for all of our kids. So this is where we need to grow. This is what our pillars need to look like. We need to have a clinical trials center everywhere that we can in order to do this. So now we ask you, how are we going to get all of this done? It doesn't just take money. That's a huge part of it, but it takes you. It takes such a large, complicated conglomerate of people to be able to work together as pharmaceutical companies, researchers, clinicians, advocacy groups, government, FDA, policy, parents, caregivers, grandparents. So many people need to be here to help get this done. And so you can help and join the Army. You can become an ambassador for science. Know the science, love the science, understand the science, and explain the science. Because the more you explain, the more people are going to know that that might be something that they may be interested in investing in. That might be something they're interested in donating to. Very, very important for our kids. Become a fundraiser. Not only to fundraise to raise money, which is obviously hugely important. We can't afford to do all of this. But eventually, with all of our help and everyone around the world and our model of bringing in funds in a really big way in order to fund really big things, we want to raise awareness at these fundraisers. Let your family, your community, your local people support you and your child to understand all the great things that are happening. But you need to be able to talk about this at these events so that they know why they're there and they want to help you. They want to support you and your family, but they need to understand it. Knowledge is power for all of us, but it's also it's power for all of them. And then become an advocate. We are starting a very strong policy advocacy program, working with government and Congress folks, and really ensuring that we have visibility, that people see Angelman syndrome, they heard of Angelman syndrome, that if there's a question about Angelman syndrome, they know who to come to, our community. And so advocacy is very important. And this is where we need to partner with all communities in Angelman syndrome in the United States and around the world to be sure that we are the best advocates and we have the best policy plan. Our FAST Advisory Council is working very hard to create a policy program. And if anyone's interested in joining this mission, please talk to Todd Werner, who's the head of our FAST Advisory Council. And I'm sure he would love to get you all involved in areas that you feel are your expertise to help the mission. So in close, there are numerous potential therapeutic platforms invested in Angelman syndrome. AS is unique with some of the most promising clinical results to date. We're focused to identify gaps and accelerated all potential strategies because we believe all shots on goal is the way to get the best approved treatments for all children, regardless of their age and regardless of their genotype, in order to change their life. It's time to create the infrastructure, not only for mice, not only for rats, and sorry, Scott, not only for pigs, but for humans that are living with Angelman syndrome. So Liz, thank you for starting the infrastructure, but I encourage anyone that wants to join and build their own infrastructure to come and talk to Liz. We're creating a matrix on how we can do this and what sites need to have in order for this to be potentially successful at their institutions as well. We are here to grow outside of Chicago. No offense, but not everyone wants to go to Chicago. We need to grow outside of Chicago, all around the country and all around the world. So there are many disorders that will not be cured or treated in our lifetime, but I am absolutely positive Angelman syndrome will not be one of them. I want to thank you all for listening, for learning, for paying attention, because remember that this is not only possible, but it's absolutely probable. Thank you.